Hello, everyone, and welcome. And today we are going to start with another subject, which is science and technology. What I've done is, if you have already enrolled for the course, you would find that we have actually uploaded two documents. I mean, you can check them out. You can check them out. Basically, what's happening is there are two kinds of document. One is your previous year questions. Okay, one is your previous year question, and another is your this document, which is your biotech part. Okay. So the point is, in today's class, what we are going to do is, yeah. So in today's class, what we're going to do is we're going to start with a very important topic, which is your biotech part. The whole point is, if you see the previous year questions, as you can see on the board in this particular document that we've uploaded, there are roughly around five questions which have been mentioned, and the weightage of these questions has increased from 2021 to 2024. Okay, even not just in forest services, but even if you look at the CSE part, which is the civil services part, you'll be finding that in CSE also pure biotechnology questions have increased. Okay, in recent years. Now, how your CSE portion is different from your forest is that in CSE they will never ask you individual technologies. Okay, they might ask you a broader question related to biopharmaceuticals or genetic engineering or sequencing, genome sequencing, a gene modification or stem cell, etc. But they will never ask you to explain a particular technology. For example, in today's class, we will learn about CAR T cell therapy, right? We will also learn about different, revival of dire wounds. So the point is that UPSC CSC paper will never ask you such individual questions for 10 or 15 mark or whatever it is. But in forest, if you look at the questions, okay, they have asked you questions on recombinant DNA technology. Okay, which is very general question that can be answered. Similarly, they have asked you question on HTBT cotton. Now, this question you cannot answer until you exactly know what is HTBT cotton. And writing 10 markers, I mean, 2 pages, you cannot fill it until you know something, right? So the whole point is that these are very, very comprehensive notes. I have personally made them, and I've ensured that nothing is left behind, even if there is like only one particular question. Okay, so from one topic, for example, like this question that was mentioned over here, HTBT cotton. From notes itself, if you study, you'll be able to answer it. Okay, so without wasting any much more time, let's just start with the biotechnology part. Now, okay, I'll give you the basics of it, and then we will just look at the PDF. Okay, now first of all, just a second. Yeah. So, first of all, what is biotechnology, right? Now, if I talk about biotechnology, you will always make these two circles. Okay. One will be your biology portion, another will be your technical portion, and then you will just mark this intersection as your biotechnology part. Okay. So, biotechnology means what? When you are using biological products, they can be living, dead, or any, living organisms. Some parts of it, okay, not like, for example, their DNA or some protein or any component of the living organism. So basically biological organisms, which can be microorganisms, plants, animals, anything, fungus, etc., or their parts, living or dead, and then you basically put some technology in it. Okay, make some newer products so that you can use them for various purposes. They can be agriculture, medical, your research and sciences projects, your space science, okay, your nuclear science, your environmental sciences. So basically, many different fields you will find your biotechnology has applications. But the whole point is that you should be able to comprehensively first answer what is biotechnology. Okay, so what is happening in biotechnology? See, in biotechnology, you are just using biological components and using technology over them to produce something which can be useful for us. Okay, now, what we listen to when we talk about biotechnology, what do we actually in our head? What biotechnology picture looks like? Mostly, we in our head it is only about medical or pharmaceutical. Okay, but also remember when we talk about biotechnology, some substances have come which can degrade plastic. Okay, some moth or some products which are made from the living plants or animals which can actually degrade plastic faster. So that will also come under biotechnology. Production of your alcohol, your wine. Okay, use that fermentation process, that is also biotechnology only. Then your yeast, production of your antibiotics, that is also biotechnology. Similarly, now you are producing stem cells, right? Basically somebody's some organ is not working, now you are thinking about using stem cell therapy to reproduce that organ, so that to produce that organ again, so that is also part of your biotechnology. Okay, similarly you will find recently there was a Nobel Prize given, okay, to two ladies for discovery of this CRISPR Cas9. So CRISPR Cas9 is nothing but a technology for genome editing. Okay, so what do we do is, suppose you all know that we have a DNA in our body. DNA is a double helical structure. Okay, and every some parts of DNA basically code for specific features, and these are genes, okay, which are inherited from one generation to another. Okay, now suppose if there's a faulty gene, you might have some disease because of it. Okay, there are some genetic diseases, right? So why are they called genetic? Because there is some coding which is ensuring that these diseases transfer from one generation to other. Okay, now if we have to change this, then we need to change this particular gene. Gene means there's some coding of some nucleotide bases, ATCG. Okay, so we have to change this particular sequence, the sequence in which these nucleotide bases, in which these nitrogenous base pairs are occurring. So when we change this particular sequence, that's how we'll be changing this particular gene. Okay, and that is how you will be repairing that faulty particular gene which is responsible for that particular disease. Okay, so the whole point is, everything that we look on the surface, somewhere or the other it is located, it is actually related to biotechnology at the micro level. Okay, so when we talk about sickle cell anemia, it is actually a disease, but is it a genetic disease? Yes. So what is happening is, there is some particular kind of coding that is present on our DNA, because of which we are able to get that disease. Now if we have to change that particular coding, we need some tool for it. Okay, and CRISPR Cas9 is one such tool which was in use recently. Okay, so okay, so now if you look at your document, we have mentioned first of all what is biotechnology, which is a pretty easy thing to know. Then methods in biotechnology that you can skip, because we will be learning about each and like each of those methods that have been mentioned in that particular document. Okay, then this can be an important question in itself, because when we look at the CSC paper, in CSC paper there was one full-fledged question on biotechnology. Okay, describe biotechnology, describe its various uses. Okay, its applications in the field of medical sciences, agriculture, and environment. Okay, some challenges that we are facing today, and also some recommended solutions. So this is the kind of question that you get in CSC, but in forest you can get a question on individual technology as well. Okay, so when we talk about biotechnology, its application, depending upon which field you are applying it to, we have given it different colors. For example, you have hydrogen, right? So we have green hydrogen, blue hydrogen, gray hydrogen, black hydrogen, brown hydrogen, etc. Similarly, turquoise hydrogen. Similarly, you will find you have different kinds of biotechnology. So when I talk about green biotechnology, it is related to biotechnology in the field of agriculture. When I talk about red biotechnology, it is biotechnology in the field of medical sciences. Okay. When we talk about blue biotechnology, it is about marine resources. It's pretty easy, right? Similarly, if I talk about white biotechnology, remember those white goods, ACs, etc. That means we're talking somewhere around industry. Okay, so application of biotechnology in industry. Okay, similarly, I mean, there are some other kinds of biotechnology also. For example, your environment biotechnology, your animal biotechnology. So now the question, obvious question that should come to your mind is, now when you talk about red biotechnology, for example, you are saying application of biotechnology in the fields of medical sciences. So what are the kinds of applications we are looking at? We are looking at everything from preparation of medicines to discovery of vaccines to basically making prosthetic legs, okay, or advanced stem cell therapy, using your CRISPR, Cas9, etc., to cure some, inheritable diseases. Similarly, your CAR T cell therapy, your gene cell therapy, okay, your genome sequencing, all like everything that you can think of in biotechnology related to medical sciences will be part of your medical biotechnology or red biotechnology. Okay, similarly, when I talk about environment biotechnology, so you must have heard about oleovorus X. Okay, so this was a particular kind of a organism which was used to basically for bioremediation, that means for cleaning of environment purposes, for reducing the pollution in the environment. Okay, similarly, basically for oil spills, okay, cleaning up of oil spills. Similarly, you must have heard about oil zapper. So these are all the organisms that you use for cleaning the oil spill because it pollutes the environment. So in environment, when I talk about biotechnology, you will be talking about new products that can be developed using this biotechnology to sort of reduce pollution, to make your environment better, or for example, your biofuels, your Jetrofa, etc., your fourth generation biofuels. So that will also be your part of environment biotechnology. Okay. Similarly, when I talk about animal biotechnology, in this you must remember two things. First, we also know that cloning happens, right? And we also know about artificial insemination process. We also know about Rashtriya Gokul mission, where government is actually actively using this biotechnology portion to preserve the indigenous breeds, to basically ensure that these indigenous breeds are actually promoted amongst farmers as well. So see how this biotechnology is not just in the labs anymore; it is also going to the farmers' fields. Okay. Similarly, in animal biotechnology, you must have heard about a project by Colosal, which is an organization where they have revived this dire wolves. Now, dire wolves, you must, if anybody of you must have seen Game of Thrones. Okay, remember that House of Stark basically adopts these five wolves, white color, like really large size wolves. So those are called as dire wolves, and they are no longer in existence. They used to exist long time back. Okay, now we have wolves which are whose ancestors are dire wolves, but dire wolves were no longer available. So what this organization has done is that they had some fossil fuels and some cells, okay, which they could take out from the very old fossils of dire wolves, and then they basically used the biotechnology, the DNA sequencing, genome sequencing, gene editing, etc., to produce exact same copies of dire wolves which used to exist long time back. Similarly, you must have heard of the project called as woolly mammoth. Okay, you must have heard of the bacteria called as Wolbachia. You can write these names down. Okay, Wolbachia bacteria. So Wolbachia bacteria, remember, uh, this was to reduce the population of mosquitoes which cause malaria, etc. What is happening is mosquitoes, right? So they lay a lot of eggs and they reproduce very very fast. So what you do is you introduce this bacteria, this Wolbachia in Wolbachia mosquitoes, which are sterile, so they cannot reproduce. So what is happening is that the current population then mates with these specifically designed genetically engineered mosquitoes, they are not able to produce offsprings. So that is how you'll be reducing the population of insects. Understood? So can you see the diversity of topics that we're going to deal today? And so the whole chapter is going to be really interesting. But before that, whenever you're asked about biotechnology, always remember to mention some few facts. Okay. Firstly, you must know somewhere a rough idea about the size of biotechnology or bioeconomy in India. Somewhere, currently it is around $80 billion. 80 or you can say it has crossed $100 billion because it was mentioned in the budget also. So somewhere it has crossed $100 billion, but the potential is three times. Is $300 billion. Okay, and our target is also to take it to $300 billion by 2030. Similarly, if you talk about because biotechnology is a growing field, somewhere India has a lot of startups, and India is doing a lot of things to ensure that startups of particular sector. Even in my college also, we had a DBT, Center for Excellence for Biotechnology. Okay, where new products are basically innovated and discovered. Now, if you talk about major in biotechnology also, there will be fields, right? For example, there will be medical biotechnology, there will be agriculture, there will be environment, there will be industrial. So major it you will find somewhere around 40%, okay, plus minus 4, is dominated by two sectors. So roughly around the environment and all other things combined. Okay, in India, always remember biotechnology. We have a special department called as Department of Biotechnology. It comes under Ministry of Science and Technology, and there are other organizations. For example, there is a PPP, Public-Private Partnership, and that organization is called as BIRAC. Okay, which started this India Biopharma mission, on which UPSC has asked a full-fledged mains question. Okay, so India Biopharma mission, which was started by BIRAC, it is a PPP, Public-Private Partnership between DBT and other private entities. Okay, so you can find in your doc some various initiatives that have been taken to promote biotechnology. For example, there is National Biopharma Mission, which I already told you has been asked by CSE, a full-fledged 10 marks question. Then there is I3, which is Innovate in India. You can write the full form if it is not mentioned in your doc. It is Innovate in India scheme. Similarly, there is Biotech Kisan that we already study, which is in combination with the Ministry of Agriculture, wherein you are basically promoting biotechnological products to be used by farmers, so giving them training, education, skills, etc. Then there is One Health Consortium, which has been active specifically after COVID-19, because we all know that how COVID-19 virus basically came from the bats, and this virus was not to be no, not supposed to be spreading in humans in the first place, but somehow it jumped that organism barrier, that species barrier, and that is why we got so much infected. Okay, and then there is obviously biotech parks, and then there are schemes for PLI schemes for production of medical devices, medical products, KPIs, etc. So they will all form a larger picture of biotechnology in India. See, I'm going to mention over here some challenges, and these challenges, if you do not remember some specific challenges related to a particular technology in biotech, you can mention these general challenges because they remain the same. In India, major challenges funds. Obviously, such high tech, technology requires a lot of funds, and we know that in India, how much do we spend on research and development? Somewhere around 0.6 to 0.7% of our GDP. That's it. If you talk about US, South Korea, etc., they are spending somewhere around 3 to 4% of their GDP, which is a good number. And that too, out of this 0.6 to 0.7%, you will find that 60% is spent by the government, 40% is only which is amount is spent by the private. Out of the 60% also, somewhere major proportion is done by the central government. State governments have not been that much active. So somewhere around, if you find, India is lagging behind in the research part. It is also lagging behind in the funds part of it. Similarly, this biotechnology has given us a lot of ethical concerns. All of these new technologies, even if you don't know anything, these are some of the challenges that are like across technologies, be it nanotech, be it AI, be it ML, be it robotics, like all of them have the same challenges. Funds is an issue, research and development is an issue, ethics is an issue. Robots, what if the robots started killing humans? Artificial intelligence, you all know the problems of deepfakes, security, safety, data privacy, right? Similarly, if you talk about biotechnology, you know the problem of designer babies. In China, there were some designer babies that were born. Basically, parents started paying to have their kid a certain way. Like they wanted eyes a certain way, they wanted lips a certain way, they wanted their child to look a certain way, they wanted their child to be intelligent, so they wanted some encoding for that particular gene. So what are you doing now? You are basically playing with the God's creation, right? So somewhere this the concept of designer babies. Okay. Similarly, problem of trip equity. See, point is all these high tech research, no, they are generally performed in the developed world. Okay, developing countries, least developed countries, they already lag behind. Then this problem of income inequality also, rich versus poor. Then problem of high class versus low class. Who will have the money to actually produce designer babies? Obviously, rich class. So what will then poor people do? Right? So this creates problem of inequality, ethical issues, right? Then there is a problem of biosafety. We all know what coronavirus, okay, meant for only lab purpose when got released in the environment, what it do. So living modified organisms, if not handled properly, can lead to risks, okay, as huge as coronaviruses as well. So how do you basically ensure that such risks are not created? Then there is a problem of unknown things. You are playing around with living organisms, okay, so you need to have some precautionary principle in mind. The problem is someday these organisms, if they become resistant, okay, they might start attacking humans only, or your creations might start attacking yourself. So how do you ensure that there is precautionary principle in place? Okay, so these are roughly the challenges which will remain the same irrespective of technology. We're talking about irrespective of biotechnology or nanotechnology. Now, what we will do is we will start with first of all biotech and agriculture. Okay, so we will look around all the particular happenings in the news recently, and some new innovations in the case of agriculture. You already had a full-fledged question on HTBT cotton. Okay, so what I'm going to do is I'm going to explain to you what is the first of all BT cotton, then what is HTBT cotton. Then there are two more crops, okay, genetically modified crops which were on the approval stage but have not been approved in India yet. One is Dhara mustard 11, okay, which is also called as GM mustard, and then there is your BT brinjal. Okay, so these are the two crops that we will be discussing, and like the entire gamut of it, what is there, what is not there in this, and how are these crops formed, what are the things that they're trying to address. And apart from that, see, in your doc, I have taken this particular information from PIB. Okay, so PIB, when you open in the month of December, they will give you update about every department, like their achievements in the entire year, and what have they achieved so far, any new discoveries, etc. So when you look at the DBT page, Ministry of Science and Technology, and in that Department of Biotechnology, you will find that there were these 9-10 points which were written in the PIB itself. Okay, the achievements of the biotechnology in agriculture. So first was development of this climate smart crop, which is very, very important. Even in this year's budget, you must have seen that government is planning to release 100 new varieties of seeds which are climatically smart, because you all know that climate change is happening. If you must have read the economic survey last year, they mentioned how climate change is going to affect the yield of all the major crops, especially rice and wheat, which is a staple diet of India. So suppose if the yield was like this, it is going to drop, okay, because of climate related changes happening. Why? Because temperature might increase, your agriculture climatic zones might change, your erratic rainfall will be there, floods and droughts will be there, right? Pest attacks might increase. So all of this is going to impact your yield in agriculture. Okay. So what do we need is climate smart crops. So here the example that has been given in your document is your, okay, it must be mentioned. We have already discussed all of this. Yeah, here you can see this is your Satvik, which is your climate smart crop, Satvik NC9. Okay, it's a chickpea variety and it is basically climate resilient crop. Similarly, you have produced some genome edited crop, basically some rice. In rice, you have changed some genes to increase the productivity of rice. Why do we need to increase the productivity? So here's a graph that I'm going to make and that you are going to make in your exam as well. Okay, so this is a bar graph and this is India, this is US, and this is China. So when I talk about productivity of rice in India, per hectare only 2000 kgs is produced. In US, you have 4000 kgs of rice being produced per hectare, and in China it is 6000 kgs of rice. So can you see productivity of which country is higher? Obviously China. So that is why India needs to improve its productivity, and hence we have come up with some genome edited crop. Then there is some genotyping arrays. Now what is this genotyping arrays? Basically, uh, like we say, right, every individual is unique and they have some unique fingerprints. So basically identifying these unique fingerprints or DNA for particular crops. Okay, then you have here you have amaranth genetic resources. Okay, some database is there, which is basically to screen the crops faster and to ensure that how you can control obesity and other kinds of diseases in humans. Then there's some fungal biocontrol solutions, and then there is some Kisan Kavach. Now, Kisan Kavach is really important because it is an anti-pesticide protective suit. You must have seen in the news also. Basically, a farmer was wearing some sort of a suit so that whenever he sprays or she sprays a pesticide or insecticide on the crops, he or she doesn't get impacted from the toxicity of the pesticide. Okay, so these are all the revolutions that have taken place according to PIB. In animal biotechnology, also they say that we have come up with better, veterinary diagnostics so that we can see what kind of diseases our animals are facing, how genome sequences, identifying some disease-resistant breeds. Okay, and I already told you that artificial insemination program. Similarly, in marine biotechnology, also, what are you doing? You're tapping the marine resources, your betterment of shrimps, better ways. How can you, keep your aquaculture, fish breeding, etc. So all of this will be a part of your biotechnology. You can read it from here, but I mean, see, the whole point is you don't need to remember all of these points together. All you need is that you identify 5 points and just revise them again and again. Okay, so that in that way you'll be able to write a better answer. Now let's come to the first topic, which is a genetically modified organisms. Now first of all, what is a genetically modified organism? See, any organism whose DNA has been altered, or genetic material. Always remember, in organisms you have two kinds of genetic material. One is your DNA, okay, and one is your RNA. In humans, what do we have? We have DNA. But suppose, for example, coronavirus. So coronavirus had RNA. Always remember, these are the only two forms of genetic material through which information gets transferred from one generation to other generation. Okay, in humans you have DNA, in virus etc. you have RNA. DNA is deoxyribonucleic acid, RNA is ribose nucleic acid. We all know DNA is double helical, RNA is single helical, which is heavily coiled. Okay, and okay. Now, when we talk about genetically modified organisms, what I'm trying to say is, suppose if this was your DNA, right? And there was some particular coding. Now that coding sequence has changed. Suppose it was A, B, C, D. Okay. Now the coding is A, D, CB. So can you see the coding has changed? Okay. So when you do this, when you modify someone's genetic material, some organism's genetic material or DNA or RNA, that is when that organism becomes as genetically modified. Now, why will you do something like this? Okay. See, you must have studied about genetically modified algae which we use in your fourth generation biofuel. Why are we doing it? So that you have more production from lesser amount of raw material, right? So you have algae which are fast growing, which can be used for production of biofuels, okay? And they're also taking in CO2 pretty faster. Similarly, you must have heard about your BT cotton. What are you saying? There was a cotton crop or there was a cotton seed. Now you changed it in such a way that an insect called as the pink bollworm, it does not impact or harms this cotton crop. Why? Because this cotton crop is now producing some toxic material which when consumed by this pink bollworm, okay, it affects its stomach, its basically gut lining, and that is why pink bollworm will no more be attracted to this cotton crop. Okay. Similarly, you must have we will be studying about this bt brinjal and HTBT cotton, etc, etc. So these are all genetically modified. Okay, will you remember now? Okay, so here, see, genetically modified organisms, they can you can modify anything. You can modify. Remember that Wolbachia bacteria. So that is also genetically modified. There was a mosquito. You modified its DNA so that it cannot produce a fertile offspring. Okay, that is how it became a genetically modified organism. When we talk about agriculture, why will you need genetically modified agriculture crops? Remember those high yielding variety seeds that we used for green revolution? What were those? Okay, those were also some varieties where some changes were made so that these varieties were short yielding, these were high yielding, they were short duration crops, okay, and they could fulfill India's food security. So ultimate aim of producing anything has to be clear. If we are making some changes in the crop, why are we making it? We are going to increase the production, or we are making it disease resistant, or we are making it climate resistant, or we are basically talking about it surviving some harsh climatic conditions, for example, drought or floods, etc, or we want to increase the nutrition content. For example, you must have heard about beta carotene rice, right? So beta carotene rice is nothing but your golden rice, in which there is high amount of this beta carotene, which is a precursor of forming vitamin A. So if you want to cure vitamin A problem, okay, which causes night blindness in a lot of people, so what you need is you need supplements. So supplements can be in the form of this genetically modified rice as well. Okay. Amazing. So now, see, whole point is you need to modify gene, right? And what was gene? I told you some specific part of DNA. Now, if you have to modify it, you need some technology to do it, right? And how do you do it? By these three technologies. Either you insert some new gene by gene gun, okay, or you use some bacteria to transfer a new gene, or you basically use some tools like CRISPR-Cas9, which can cut a particular portion, then insert some another gene or base and then stitch it back. Okay, so here are some examples which I think are important from the perspective of your if the question hits on GMO, then you can write these examples and score more marks. Okay, BT cotton we've already discussed. See, I'm going to show you on the whiteboard what is happening is, suppose this was a cotton crop. Okay, there was a particular gene. Now what you did is you modified this gene. Okay, and there was this bacteria called as BT. Okay, what is the full form of BT? It is Bacillus thuringiensis. Okay, this was a bacteria which is found in soil and which had this particular property that it used to produce these toxins. Okay, and when it was impacted by some another harmful organism, it used to produce these toxins which used to kill the gut of that attacking organism, and that is how it used to save itself. So we use the same technology in our cotton crop also. We modified its gene so that it can this cotton crop can be protected. And this gene, okay, was your cry1ac. Cry1ac. Okay. So cry1ac gene is the gene which is responsible for providing pink bollworm resistance to our cotton crop. Okay. Almost in India, 98% of the area which is under cotton cultivation is now under BT cotton. Now the problem was this BT cotton crop. Okay, we used to okay, this cotton crop. There were some other crops also that used to grow alongside cotton. Those are called as weed. Okay, weed is not that weed, the drugs that you're thinking about. Weed is basically any unwanted plant, any undesired plant which grows in the same space as your crops. It competes with your crop for food, water, shelter, nutrition, etc. So ultimately, what's happening? Your production and productivity of your main crop is reducing because of the presence of some other kinds of crop, which is your weed. Okay, so now to remove this weed, either you can do it manually or you can spray some sort of solution which will kill this weed, right? So those materials which kill the weeds, they're either called as herbicides or weedicides. Okay, so they're called as herbicides or they're called as weedicides. Okay, so what was happening in our cotton crop, we used to basically put lot of herbicide, okay, to kill these excess of weeds. Now the problem is, because of this, obviously, because you're spraying some toxic material, your cotton production productivity gets impacted, your good bacteria or good organisms also get killed in the process. Third, you need to spend some extra money to buy those insecticides and pesticides and spray, sorry, those weedicides and herbicides, and then spray on your crop. And then your farmers also need some protective gear. So a lot of issues are there because of this weedicide spraying. So now we came up with what? We came up with HTBT cotton. Now this beat first there was some cotton in which what you did, you basically modified its gene to produce BT cotton. And now in BT cotton, you again inserted some new gene so that you can produce HTBT cotton. Now, what is HTBT cotton? Its full form is herbicide tolerant Bacillus thuringiensis cotton. So, BT cotton, which is made now herbicide tolerant. So, even if you spray herbicides on it, your cotton crop will not get impacted, only your weeds will get impacted. Okay, and there will be less of weed growth also. So, this is your HTBT cotton. Similarly, your golden rice I already told you, basically vitamin A deficiency. Dhara mustard 11. Okay, I'll explain it to you. See, okay, we'll do one thing. We'll learn about Dhara mustard 11. I think the whole chapter is there on Dhara mustard 11. And then there is BT brinjal. Okay, this is one part of GMO application in agriculture. Then other part of application of GMOs must be in medicine. So how will you use these genetically modified organisms? You can use them to produce insulin. There are bacteria, right, which produce insulin. So you can produce vaccines from these bacteria, gene therapies to treat diseases. Similarly, in industry, I already told you, fourth generation biofuels, biodegradable plastics. These are all the examples. Advantages, disadvantages, concerns, challenges will always remain the same. Advantages of GMOs will be what? See, if you do not remember exact, always remember, start remembering the applications and then just put one example as your advantage. For example, advantage in biomedical sciences, advantage in agriculture, environment sustainability or reducing pollution, like that. Okay, curing genetic diseases. Okay, finding solutions, reviving your lost microorganisms or lost organisms. So these are all the examples of your GMOs and their applications. So if and their application and their advantages or basically significance. So that is what I'm telling you. You don't need to learn anything. Okay, all you need is some little bit of here and there, some you can say wisdom, and you can apply the same knowledge in multiple questions. When you talk about advantages of GMO, I already told you. Just remember the different areas where it is used and start mentioning that particular thing. Concerns will be again the same biosafety concern, same biodiversity problem. See, why am I talking about biodiversity? Is I told you that 98% of our area under cotton cultivation is now under BT cotton. Now tell me, if there is a particular kind of a disease which BT cotton is hit by, don't you think 90% of cotton will get destroyed because it does not have biological diversity? Have you heard about Irish potato famine? What was the problem? Problem was that in Ireland, they were growing a certain species of potato, okay, or they were growing certain kind of potato, and those potatoes were genetically similar to each other. So when a particular pest attacked this potato, entire nation's potato crop got destroyed because they had no particular biodiversity. That's what I'm telling you, we need biodiversity as well. Okay, and then obviously, because India is a growing country, so always remember we have some regulatory issues. Okay, if you talk about globally, this acceptance of GMO, in India, the only genetically modified crop which is approved is GM cotton. And always remember that in India, no edible crop has been approved so far. So through cotton, what you do, you basically make products, right? Your textiles, but you don't eat cotton. So in India, you have not approved any of the uh, edible crops. Okay, while in US you will find genetically modified soybean, genetically modified alfalfa, genetically modified maize. Okay, in Bangladesh also brinjal etc. is there. So there are many countries in the world, for example, USA, Brazil, Argentina, Canada, China, which are heavily dependent on these GMO crops. In India, only BT cotton. Two more crops were in approval stage but not yet approved. Okay. Now let's come to the first stage, which is your BT cotton. I've already explained it to you. Bacillus thuringiensis, Tri1AC protein, or the gene that we're trying to change. Okay, and how basically it kills that bollworm. Okay, in India, remember, see, India signed CBD, and in CBD, which is your United Nations Convention of Biological Diversity, which came around in 1992 under Rio de Janeiro, Earth. Okay, here in 1992 there was a summit, right, which was United Nations Conference uh, for Environment and Development, which took place in Rio de Janeiro, Brazil. There were three independent bodies that came around, or conventions that came around. One was UNFCCC, another was UNCCD, which is desertification, and another was UNCBD, which is Convention to Biological Diversity. Within that, in CBD you have three things. You had Cartagena Protocol and biosafety. From biosafety, from what? Your living modified organisms. You study all this in your environment portion. Similarly, you will find you had Nagoya Protocol, which came around in 2010 for equal access and benefit sharing. And then you had Aichi biodiversity targets, which have now been replaced by Kunming Montreal biodiversity framework. Okay, don't worry, even if you don't know all this, we are going to study in the environment portion. Okay, so Aichi biodiversity targets were only there till 2020, and now new targets are in place, which is your Kunming. Kunming is a city in China, Montreal is a city in Canada. So Kunming Montreal biodiversity framework. Okay, in which we have goals to be achieved by 2030 and also till 2050. Okay, understood? So when we talk about BT cotton, remember that India signed this CBD, okay, in somewhere around 2000. Our Biodiversity Act also came around in 2002. This is when India formed this, India already had this body called as Genetic Engineering Approval Committee, okay, GEAC, which is the body which is responsible for all the genetically modified products or crops which are grown in India. Okay, so remember that in GEAC, it comes under not Ministry of Agriculture, it is under Ministry of Environment, Forest and Climate Change. Okay, so you will find that India, the first crop that got approved of genetic modification is your BT cotton. Okay, so the first crop that got approved was BT cotton, and when did it get approved? It got approved in 2002. Okay, so this is your BT cotton, and I already told you. And okay, see, this is something I'll be telling you when we talk about crops. So remember the challenges that we had, those were the common challenges. Along with it, mention that how majorly whatever genetic modification is happening, these crops are getting developed, or these new seeds are getting developed by these private companies like Monsanto. Okay, and that is why these companies charge exorbitant prices to farmers. Secondly, these seeds, which are genetically modified, they are not capable of farm saved seeds. That means you cannot save and then produce it in the next agriculture year. You have to, every time when you're thinking of cultivation, you'll have to buy fresh seeds from the company. Remember how our farmers basically they grow crops, okay, then they harvest them and then they sell those crops in the market, but they also keep some seeds for growing those seeds in the next agriculture year. But with these seeds, which are genetically modified, you cannot farm save them. Okay, you have to buy every year from these big companies and MNCs, which are ultimately exploiting the farmers. Okay, remember? So this is the uh, issue with when I talk about these genetically modified crops, for example, your BT cotton. Benefits, obviously, India became self-sufficient in cotton. See, whenever you talk about benefits, why are you even thinking about producing genetically modified crops? A, obviously, production might increase. B, productivity. some resistance might be developing, okay, because of which your insect attack is reducing, farmers expenses on insecticides are reducing, because of which what is happening ultimately, farmers are saving more money, they can utilize that money to build some infrastructure in their own farm. So you can just keep connecting on the advantages if you just identify the core one or two advantages, okay. Similarly, see here, pesticide uses is reduced, you have better yields, you have economic gains for farmers because now they are saving more income, and also some positive environment impact because we are not using that much of pesticide. But obviously, challenges remain. Now let's talk about HTBT cotton. See, HTBT, I already told you, you had BT cotton, and now you wanted to reduce the weeds, the undesirable plants that were growing in your alongside your crop in the farm field. So basically, instead of now, spraying herbicides every time, you produced a cotton variety which is a herbicide tolerant, okay. So what is happening is you had BT cotton which was insect resistant or pink bollworm resistant, and now you had HT cotton also, which is basically protecting your crops against your herbicides, okay. And what will happen, you don't have to spray again and again, so ultimately benefits are the same, only what you studied in BT cotton, just the wordings will change, okay. So claim benefits, I already told you, weed management, you don't have undesirable plants, so production, productivity, yield, all that. Labor cost reduction, again and again you don't have to spray, so you're saving not only on insecticides, pesticides, weedicides, etc., but you're also saving on that spraying cost, your labor cost, because now farmers can work. If they were taking 2 days on the field to spray or herbicide, now they are free for those 2 days, right? So they can provide their labor in some other farmer's field and earn some more money also, and concerns are the same only, okay. Then this is important, please star market, because it can be asked as an individual question as well. Now is your BT brinjal. What is a BT brinjal? See, BT brinjal again. You know what is BT cotton? Bacillus thuringiensis cotton, right? So basically, it was protecting your crop against pink bollworm. Now, if you talk about brinjal, you will find that brinjal has some leaves, then it has some bengan or brinjal, and then there is roots. Brinjal plant attracts this fruit borer. Okay, remember this insect name, because until and unless you write this name of the insect, you won't get full marks. Okay. So basically, what's happening is this BT brinjal, no, BT brinjal attracts this borer. Borer is nothing but insect, okay, which basically eats on your fruits. So this brinjal, right, bangan that you see in your houses. So there is some insect that eats that bangan, which is your fruit and shoot borer. So to save a crop from this fruit and shoot borer, which is actually causing a lot of losses to the farmer, you came up with this BT brinjal. Again, what will this BT brinjal do? This BT brinjal will basically produce these protein, because Bacillus thuringiensis, again it will produce some cry protein, cry A1AC protein, okay, and this protein is toxic to this borer. Why? Because it will kill this borer, okay, and that is how your plant will be saved. I have written the events about BT brinjal, timeline in India. You don't need it actually, but remember, for BT cotton, which company was responsible? Monsanto, which is a company from US. Now BT brinjal is an in-house production. It was developed by Myco, which is your company cooperative in Maharashtra. Okay, so Myco, and in collaboration with some public institutions. Okay, it has not yet been approved for usage, but it is said that in India, many states illegally BT brinjal is being produced by farmers. Okay, obviously benefits will be what you will be saving your farm, your brinjal crop from fruit and shoot borer. So obviously production, productivity, yield. Okay, then you don't have to spray insecticides, pesticides. There will be no residue also. There will be no pollution also, soil pollution, water pollution, air pollution, emissions, all that. Then you'll be saving on spraying, so the labor cost is also saved. Your buying of insecticide, that part of money is also saved, right? So all that is there. What about the concerns and controversies? Same thing only. Problem of health, problem of biosafety, precautionary principles, environmental concerns. Then similar crop, for example, now you don't have any variety in cotton. 90% of your cotton crop is same. Similarly, you will find the problem is in BT brinjal also, because eventually farmers will start adopting only this particular brinjal, and you will not have any variety. So problems like Irish potato famine can happen in India as well. Okay. Now let's come to Dhara Mustard 11. See, you don't have to read or go through the document word by word. All you need is just these headings. And if see, okay, I'll just show you. Point is, for a 10 marker question, you have these two pages. Okay, all examiner wants is first of all that all the parts asked in your question should be answered, and try making your answer point wise. Okay, so that it's easy to read, it's easy to identify what is the heading that you're addressing, and it's easy to check also. Okay, so when I talk about BT brinjal, what uh, things or what particular topic that or heading that should come to your mind, what are the topics that should come to your mind? First of all, what is BT brinjal? Then always in agriculture start making diagrams. Okay, just a basic diagram will work. For example, something like this. Okay, or you can make a normal plant and then show some bangan or some brinjal on it, and then mention some insect, and then just name it as fruit and shoot borer. Here, write brinjal. This is how you will get marks. Okay, whenever there's a scope of making a diagram, please make a diagram. Okay, so in BT brinjal, there's diagram. Then there is what is Bacillus thuringiensis brinjal. Then how is it made, and then of course its challenges, its benefits, and way forward. Okay. Now let's talk about Dhara Mustard 11. Please star mark it. Dhara Mustard 11 again is nothing but GM mustard only. Okay. And this is also produced by Delhi University. So it is also an in-house production, unlike BT cotton, which was by Monsanto. Okay. Here, remember this word, and you have to mention this word, which is your Barnes and Barnstar genes. So these are some particular kinds of gene which provide this particular property to make mustard your GM mustard. Okay, what is happening is, see, okay, I told you, right? DNA has a lot of genes in it. So suppose there is a gene. Okay, if it is present, your the plant, uh, the plant becomes sterile. Plant is male sterile. Okay, so that particular gene is your Barnes. Similarly, there is another gene which restores your fertility. Okay, that is your Bard star. You don't need to go in depth of it because you wouldn't be needing it. I mean, it's important for botany, agriculture engineering, and all those optional students, biology, but it is not important for you. Or just remember, in GM mustard or Dhara mustard 11, you have two kinds of genes, which is Barnes and Barstar. Barnes is the one which is your male sterile gene, and to restore that fertility, okay, to ensure that the crop is fertile again, you have this Barstar. Okay, and using these two particular gene combinations, what you do is you produce a hybrid variety. Okay, why would you want to produce mustard? In India, you already know that almost 50% of our edible oil is coming from outside. We're importing this edible oil needs. What we are importing is palm oil. Palm oil is coming after destroying these Indonesian forests. In India, the majority of edible oil that we produce is either sunflower, okay, or your mustard oil. So mustard, to improve the production yield and productivity of mustard, you have this crop of GM mustard. Understood? Now, obviously, benefits will be what? Benefits will be again the same. You have higher yields. See, even if you don't remember this figure now, this figure is a very safe figure. You can mention anything between 15 to 25. Okay, because I mean, if there is a minute change in a particular thing, 15 to 25% yield is pretty normal to achieve. Okay, so higher yield in the form of 15 to 25%. Then you have obviously because you will produce more in-house oil, so you will reduce your forex reserve bill also. You will be saving on that import bills also, and also since it's produced by Indian university, so you will be providing its license to produce commercially to some Indian company only. So that way, more employment generation as well. Okay, again, I have given you the timeline, not needed as such, but remember that in India, this crop again was in trials only, but has not yet been approved for consumption purposes. Concerns again the same, so I'm not going to discuss it again. But the whole idea you understood. Okay. So here what we've done is we discussed four crops so far: BT cotton, HTBT cotton. Then you had your Dhara mustard 11, which is your DMH 11, and your BT brinjal. Okay. Now there is this word called as gene revolution. This was something which was in news, and I thought this can be asked as 10 marker in forest services. Now, what is a gene revolution? Okay, idea is that there are two gene revolutions that have taken place. First was in 1970s, the time when your green revolution was happening. So you were inserting new genes, genetically modifying. Uh, okay, if you don't remember the name, remember this variety called as Sonalika, okay, and Kalyan Sona. So you have your normal wheat. In that you modified their gene, and the two new variety of wheat, they were called as Sonalika and Kalyan Sona. Similarly, when you talk about rice, okay, what was the gene that you inserted so that you had better production, short duration rice crop, etc. So this was DG, Wu Gen gene. Okay, you don't need to remember it, but DG Wu Gen is gene. Okay, so this is the gene that you inserted in the rice crop to make it more productive, to basically use it in the green revolution. Okay, so first gene revolution happened back then. Okay, now second gene revolution is using these techniques, specific techniques called CRISPR-Cas9, which is basically for your gene editing. Okay, which allows for, see, whole idea is genes are encoded at specific places. If you don't have specific or precise cutters, okay, or those tools which can modify genes at specific locations, then you will obviously be making some undesirable changes. So CRISPR-Cas9 is a technique which is capable of making these specific changes. Okay, so with this, what we have done is we have completed biotechnology in agriculture, specifically some new technologies which were in news. Now we jump to the next part of it, which is very, very interesting and I think more relatable also to you guys, which is nothing but biotechnology and biopharmaceuticals, basically your medical industry. Okay, now, when I talk about biopharmaceuticals, again, the broader topic, okay, broader topic will remain same. First of all, what is biopharmaceutical? You make these two dabbas. One is a biology, another is a pharmaceutical, and then intersection portion is a biopharmaceutical. Okay. Now, what is a biopharmaceutical? Again, when you're using these living modified organisms, your technological products made from biological organisms, okay, or their parts, or their or some products which are made from those living organisms, and you are using them in the medical field, then that becomes your biopharmaceuticals or biotechnology in medical sciences. Okay, now when I talk about biopharmaceuticals, how is it different from the normal pharmaceuticals that we see around? See, when you talk about normal tablets, I mean, crocin is not specific to one particular person, right? But when I talk about biotechnology and pharmaceuticals or biopharmaceuticals, you must remember that this is very specific technology. It can cure personalized diseases also. A disease that might be happening, it might be so rare that it might be happening only to 1,000 people or one person in 1 lakh, so that those diseases can also be cured using biopharmaceuticals. Okay. Now these are few names I want you to have a look at. One is your monoclonal antibodies. Okay, what are antibodies? When I talk about a blood group as A, B, okay, that means my body has antigens and my body has antibodies. Antibodies are the firefighters of the body, right? So basically, these are the molecules. When some infection is happening, these are the molecules. Antibodies must be there to fight that particular infection. So when I talk about monoclonal antibodies, I mean they are used for curing cancer or autoimmune diseases. When I talk about recombinant proteins, whenever you hear the word this recombinant, it just generally means a DNA which has been modified. Okay. So your recombinant proteins means proteins which are formed after modification of DNA. Okay, so you can use them for production of insulin, curing of diarrhea, sorry, diabetes and anemia. Similarly, vaccines you already know, Zycovid D, right? So vaccines produced, RNA vaccines, mRNA vaccines. We've already seen them during the coronaviruses. Similarly, gene therapy products. This is very important. Remember this name, Zolgensma. Gel. This is Zolgensma. Okay, so Zolgensma is basically a gene therapy product which is used for treating genetic disorders like thalassemia, hemophilia, etc., or color blindness. Then there is cell-based therapies, CAR T cell therapy. I already told you. Okay, I'll explain it to you over here only. See what is two, three concept. I'm gonna explain it to you over here. One is your three-parent baby. Okay, another is your CAR T cell therapy, and then I'm going to explain to you stem cell therapy. Now, first of all, what is three-parent baby? As the name suggests, that means this baby has three parents. But you should be like, ma'am, how can we have three parents? We can only have two parents, right? One male and one female. Actually, what happens is when I talk about DNA or genes, okay, I talk about a cell and I talk about nucleus. And I've already told you that in that agriculture class that how your nucleus has DNA, right? It has chromosomes and chromosomes have DNA. But there is this mitochondria also, which also has some circular DNA. And this circular DNA is only coming from your mother. It is not coming from your father. Okay. So this DNA, which is found inside your mitochondria, it is called as mitochondrial DNA. And the other name of it is also called as organellar genome. What is it called as? Organelle or organellar. Okay. And genome. Now, why am I telling you this? Because there has been a full-fledged question on organelle genome or organelle genome. So, genome is basically nothing but entire genetic material present in an organism. And organelle genome means the genome which is not present in nucleus but outside it. So, in plants, it is present in plastids, and in animals, it is present in the form of mitochondria. Okay, so mitochondria has DNA inside it. Now, what is happening if suppose this DNA, which is present inside the mitochondria, is faulty? So, what will happen is when you talk about fusion, the process of reproduction, male gametes, female gametes, your sperm and your ovum combine to form a zygote or an egg, okay, for that matter. Now, in this egg, half of your nuclear genes are coming from male, half of your nuclear genes are coming from your female or your mother, but entire mitochondrial DNA is coming only from your mother. So if this mitochondrial DNA is faulty, it is going to be present in your offspring also. Okay. So how do you remove this? You basically have this concept of three parent baby. And why am I teaching you this? Because in UK, this three parent baby, this entire technology has become legal. So what's happening is you have a mother, okay, whose mitochondrial DNA is faulty. Okay. There's father, they fuse together. You take their nucleus. Okay, you take their nucleus, and then there is some donor mother. You remove her nucleus, insert this nucleus inside it. Okay, so that is how the offspring that will be getting produced will have your same DNA as your mother and father, which is 99.99% DNA will be same. Only some part of this mitochondrial DNA, okay, will be from your donor mother. Understood? So three parents are involved, two your original parents, and then third is your donor mother. Okay. So within this three parent baby also, there are two kinds of techniques. One is your spindle transfer, and another is your pronuclear transferase. Okay, or nuclear transfer. What is happening is, see here, what was happening? You have only one fusion, right? And then you are replacing the donor mother's nucleus by this good nucleus, okay, which is coming after fusion. So this technology is called as spindle transfer, okay. And then there is another uh, technology within your three parent baby. What is there? This is your father, obviously, who is not faulty. Then there is your mother, okay, and then there is your donor mother. You basically fuse together father's sperm with your donor mother's ovary to basically get an donor mother's egg to produce a zygote. So you have one zygote over here with a nucleus, another you produce, your father and mother produce. Okay, now what you do is to basically remove this nucleus and insert this nucleus. But here, as you can see, that two times fusion is taking place between your original father mother and your father and your donor mother, and then one nucleus or one zygote is getting destroyed. So such technology within three parent baby is your pro nuclear transferase. Understood? This is called as pro nuclear transferase. Okay, so this is your overall technology when it comes to your three parent baby. Now let's talk about CAR T cell. Now what is this CAR T cell? Okay, this is chimeric antigen receptor T cell theory. Now you all know that in our body we have something called as T cells, we have B cells. Okay, what are B cells and T cells? These are basically your these are your immunity providing cells. So whenever you get infected by a disease, these are the cells which come and fight that particular disease. Okay, so what happens is sometimes we will find that our body, T cells, which are responsible for fighting cancer also, because it is also a disease only. Now, what is happening is that T cells are not enough, they don't have enough strength to fight the cancer. So now this is this another therapy called as CAR T cell therapy, in which what you are doing is that in CAR T cell therapy, you are modifying an organism's T cells itself. So you basically take out blood, you separate T cells from it, you modify those T cells, okay, and then you put them back in the blood, and then you insert the same blood back in the organism's body. Now in this organism's body, your T cells have been modified, and then they will be fighting cancer on their own. You don't need to put now radiation, chemotherapy, etc. to fight cancer, because your T cells have been modified to kill that those particular cancer cells. Now, first of all, what is a cancer? Tell me that. See, cancer is nothing but when your cells, okay, your cells start reproducing uncontrollably. Okay, your cells start reproducing uncontrollably. That means there is division happening, and there is no control that you have over the cell division process. So that means there will be lumps formed in certain areas of body, and that is what we call as cancer cells, okay, or cancer. Cancer is nothing but a uncontrolled division of your cells in the body, and this is harmful. Why? Because obviously it's going to form lumps, and it is going to affect the functions of those particular system wherever those cancer cells have been developed. Okay, so what these T cells will be doing? These T cells will basically be modified to kill the cancer cells on their own. So this is nothing but your CAR T cell therapy. Okay. Similarly, you will find there is something called as genome sequencing. Okay, CSIR and Ministry of Science and Technology, basically Department of Biotechnology, they're all running projects to identify the genome sequencing of Indian people. Why Indian people have a particular skin tone? Why are they, sharp or not sharp? Why a particular region's people look like that? Okay, so those are something which are encoded in our genes. And if you can read that genetic sequence of my sequence will be different from your genetic sequence, but in general, the people living in plain, okay, they might have a certain kind of genetic sequence. People living in mountains will have a certain genetic sequence, okay. So through genetic sequence, okay, the order of these nitrogenous base pairs, ATCG, through that you can identify basically why are particular characteristics found in a particular population. So CSIR, DBT, they're all running different projects for this particular identification of genome sequencing. Okay. Similarly, you must have heard about stem cells. Now, what are stem cells? Okay. You know that in our body, okay, whenever this new zygote is formed, okay, or just the fusion part of your egg and your sperm, so that zygote is formed, right? So have you ever imagined that why this particular zygote, what is it? It is nothing but combination of two cells. It is also a cell in the beginning, and then how a zygote forms an entire individual. From here only, from these cells only your heart is developed, lung is developed, your limbs are formed, head is formed, brain is formed, heart is formed. That means these are the cells which can develop into any other kind of cells. Cells which are found in liver, cells which are found in hand, cells which are found in heart, cells which are found in lungs, etc. So that means those cells which are capable of developing into other kinds of cells as well, those are called as stem cells. Okay. And stem cell therapy has become an important part because through this you can actually cure a lot of diseases. Okay. So I think we'll go back to the talk and see what is written and what do we have to discuss now. Uh, okay. So here we were talking about biopharmaceuticals. Okay, in that I gave you some five, six technologies or types of biopharmaceuticals that we are finding these days. Similarly, there's concept of biosimilars. Now, what are biosimilars? Biosimilar is nothing. For example, you have crocin. So instead of taking crocin, you have some biologically similar product to crocin, which is less harmful and obviously more cost effective also. Okay. Now tell me one thing. Since uh, so much time I'm telling you that you have to modify your gene. Okay. You have to identify your gene. You have to modify your gene. So how are you going to do it? So either you use technologies like CRISPR-Cas9, okay, or you basically insert gene using gene gun, okay, or you basically encode for that particular gene using some protein sequence. So there are a lot of ways you can do it. This is not important from your forest perspective, but what is important is this applications of biopharmaceuticals. So I already told you about cancer treatment. I already told you about producing insulin. I already told you about autoimmune disorders. Just learn the examples of autoimmune disorders. Then genetic diseases, curement, and COVID-19 mRNA vaccines. Remember, Moderna vaccine. So that is the example. Advantages, obviously, if you produce something using this specific high-end biotechnology, I mean, obviously the treatment can be personalized, it can be made more cost effective. Through this, you can identify that why only a particular population gets ill, or why a particular population is more susceptible to diseases than others. So these are the advantages. Challenges, again, by safety, funds, functionaries, regulatory balances, etc. If you talk about biopharmaceutical industry, very, very important. Just remember these 4 points, that India, you already know, is the largest producer of generic medicines, and almost 50 to 60% of generic medicines are produced in India itself, of the world I'm talking about. Okay. COVID vaccines also, remember that how we supplied not only COVID vaccines, but HCQ tablets, which are used for curing malaria. We provided them to Brazil also, and that is why Brazil's prime minister basically called India as this operation is Operation Sanjeevani, because India was providing Sanjeevani like Hanumanji was providing it to Lakshman. Okay. Then there are some major industry names that you can see. Reddy's, Biocon, Serum. I think we don't need to do that. But what we need to study is for sure this biopharmaceutical part. Okay. So in biopharmaceutical, see, significance again is the same only: treatments, diseases, personalized medicines, obviously, economic impact, these have all remained the same. Challenges, again, the same. Uh, Gene editing. What is gene editing? It's the same only. I mean, not only you can modify, you can remove a particular gene, you can add some another gene to it, you can modify the existing gene, whatever it is. So these all processes where altering the genetic material, altering the DNA, then it is called as the gene editing. Now, how will you do it? You need some tools to do it. So the Talents and ZFN. Okay, these were the tools which were used in earlier times. And now, which tool are you using? You're using CRISPR-Cas9. Okay, what will you use by editing genes? Start talking about different fields: agriculture, healthcare. I don't need to go and repeat the same thing again. Animal husbandry, conservation biology, I already told you. Revival of dire wolves by colossal. Benefits again the same, challenges again the same. See, the point is, why have I written this in this document again and again, benefits and challenges? Because there are some 2, 3 points which are different. Rest, everything remains the same. Okay. So if you just want to, minimize the effort which you are putting in this GK subject, all you can do is you can just learn the common challenges and use those challenges, significance, benefits everywhere. Okay. If you have to go and read specifically, then I've already mentioned over here specific challenges and benefits also of each of these technologies. But as far as GK portion is concerned, I think common challenges will do for you. Okay. Let's come to the most important technology, very, very important, which is your CRISPR and CRISPR-Cas9. Basically, you can okay. First of all, before we move forward, what is the full form of CRISPR? It is clustered, regularly, interspaced, short, palindromic repeats. And what is Cas9? Cas9 is nothing but a molecular scissor. Suppose this is DNA and you are asking me to modify it. I will ask you, where do you want to cut this DNA, or where do you want to modify this DNA? Okay, you tell me, okay, suppose this is DNA, and you tell me, ma'am, that I have to modify this DNA at this position. So this Cas9 is nothing but the molecular scissor. This is a protein, okay, which will help you identify, which will help CRISPR to identify the spot where DNA modification has to be made. Okay, so Cas9 is that protein which is identifying, which is binding itself. It's like this binder. It binds itself to the DNA so that position can be identified where your DNA modification or gene modification has to be made. Okay, so it is a gene editing technology, precisely cutting and modifying DNA. And you know, okay, you need to observe something, right? Either you discover it or you observe it, or this thing was already happening in some bacteria or in some organism, and you basically said, okay, why can't we use this in humans? So basically, there were two women, okay, uh, Charpentier and Doudna, which basically discovered CRISPR-Cas9 in bacteria. So bacteria, what was happening? Suppose if their DNA gets infected with some virus, they cut this particular DNA part which is infected, and then they stitch back the remaining DNA again. So, and how do they do it? They do it using this CRISPR-Cas9 mechanism. Okay, which basically cuts the particular portion of DNA precisely at the place where the infection has happened. It removes that particular, DNA segment or part from the entire DNA, and then stitches the remaining DNA back. This was observed in a, I think it should be mentioned. Wait. Yeah, this was observed where it was observed in a bacteria. Okay, it was its natural immune system. And now, when you observe this in bacteria, you said, why can't we use this in humans? So, what I've done is, forest services, they can ask a specific question on how CRISPR works. So, this, if you can make a diagram, it's really, really amazing. All you need to do is you can just even make this DNA and just write 1, 2, 3, and 4. First point is, see, there is a Cas9 protein, okay, which will form a complex with some RNA. Okay, okay. Just give me one more page. See what is happening. You must have heard the process of DNA replication. Okay, DNA replication, where suppose there is one copy of DNA and now you want to make another copy. Okay, so what happens is that first of all there is decoupling or decoiling of this DNA. So these strands which are heavily coiled and intertwined with each other, now they basically become separate. Okay, now there is some RNA portion which gets attached to this and it starts forming the other strand. Similarly, here also there is some RNA portion which gets attached to it and then it starts forming another strand. So this is how your two DNA will be formed from one DNA. Okay, this is the process of your DNA replication. Similarly, when you talk about CRISPR-Cas9, what is happening is if you have to suppose modify this portion of DNA, your Cas9 is the protein. Okay, it binds itself to this RNA complex, and this RNA complex is nothing but it binds to this DNA segment. Okay, it is the complementary DNA segment. Suppose if here A is written, then there will be T in this RNA complex. Okay, similarly, if there is C, then this will be binded to G. Understood? So, this is how you will identify this particular site where gene modification has to take place and using Cas9. Okay, and CRISPR will ultimately do its work. So Cas9, what does it do? It forms a complex with RNA, then it attaches itself to the matching DNA where the modification has to take place, and then finally your Cas9 protein, it cuts the DNA from the part where your modification has to take place. It modifies the DNA and finally they stitch back together. Okay, even if you are able to explain this much, trust me, in your GK paper, you will get really good marks and you have high chances of getting an interview call as well. Okay, significance again, when such cutting edge technologies get developed, what is the significance? Obviously, it is very, very precise tool for cutting DNA. Earlier, we had talents and ZFN, but they were not very precise. So sometimes what was happening, because they were cutting DNA in the wrong position, there were some side effects or negative impacts which greatly reduced when we are using CRISPR-Cas9. Efficiency is much faster, more effective, versatile also because it can be used in plants, animals, microorganisms, and I already told you Doudna and Charpentier. They won Nobel Prize in chemistry for discovery of this CRISPR-Cas9. Okay, CAR T cell therapy. If you even if you don't remember, just remember CAR. That means cancer. So cancer treatment of cancer using T cells. Okay, it is self-explanatory. I've already explained you the process. See here, what is happening? This is the human, which is suffering, who is suffering from cancer, and you need to basically cure cancer. What you can do is either you can put them under chemotherapy or you can use this CAR T cell therapy. So in CAR T cell, what is happening? You take out the blood, you extract white blood cells, within that also T cells. Then these T cells are modified using this CRISPR-Cas9, etc. Okay, and you are introducing these new genes called as CAR genes or CAR proteins. Okay, what these are basically antigens, and they give these T cells the power to attack cancer cells. Okay, they are engineered, they're modified. Now again, what will happen? You will fuse this blood in the organism, and now in that organism itself, those T cells will be fighting against the cancer. So you don't need to give them any external radiation or chemotherapy. So obviously much faster, much efficient, very less side effects. Okay, also might be able to cure those cancers where chemotherapy, giving chemotherapy is dangerous or expensive also. Okay, for example your brain tumors, etc. So now CAR T cell therapy, okay, first of all, why are we discussing? Because in India there is this immunoact, let me see, yeah, here in India, first indigenous CAR T cell therapy was developed by this IIT Bombay's startup and Tata Memorial Center, NextCAR 90. And now this has been approved by DBT as a legit method for curing cancer. Currently, it is only, curing leukemia, but it can be soon expanded to cure other kinds of cancers as well. Okay. So I've told you how this process works already. Okay, its applications I told you currently only for curing leukemia, but in future for curing other kinds of genetic diseases as well. For example, your lymphoma, your autoimmune diseases, etc. Challenges again same. Costing is really really high, expensive. Not everybody can afford it, and it's a cutting edge research also. What if those T cells start attacking good cells also, apart from cancer? Similarly, there might be a problem of cytokine syndrome. Now, this is something you must have heard in COVID also, that when people were given these COVID treatment, some of them basically developed this cytokine storm. Okay, and because of which your immune cells started attacking your good cells in the body, not just the infected COVID-19 cells, okay, but your good cells in the body, because of which you will find ultimately the person died. So basically, it's sort of triggering of autoimmunity inside the body itself, wherein your body is acting against your own body only. Okay, so cytokine release syndrome or cytokine syndrome is another problem. Obviously, I mean, such expensive technologies require special skills, funds, functionaries, skills, and where, I mean, you need to give proper training, etc. Labs also, regulatory structure also. So that all is missing in India. Now, here is a short comparison which can be asked in your forest services also, which is basically a difference, because these are the two kinds of treatment for cancer. So the difference between CAR T cell therapy and chemotherapy. See, in chemotherapy, what you're doing is you're using radiations to kill the particularly cancer cells. In CAR T cells, you're using body's own immune system to kill the cancer. It is obviously this CAR T cell is going to be much more targeted, much more efficient, and having less side effects as compared to chemotherapy, right? Similarly, you will find that in CAR T cells, and see, in chemotherapy, you must have heard, even if you have seen or heard somebody who is undergoing cancer treatment, they must have told you that how uh, chemotherapy sessions that they have to take. In CAR T cells, you can actually modify in one or two times only the T cells of the body. Okay, and because you're modifying someone's own T cells, that means you can give much more personalized treatment in CAR T cell therapy as compared to your chemotherapy. Now, there is another concept which is called as gene therapy. Now, what is this gene therapy? Basically, what is happening? You just decipher the words. See, in science, nothing is hard if you can decipher the words correctly. If you know exactly, okay, what this is talking about, can't you write 2 pages or 3 pages of it? Okay, here. What is gene therapy? Divide the word. Gene and therapy. Gene obviously is that particular coding which is present on your DNA, which is giving you a special character which is inheritable or transferable from one generation to another. So this is gene. Now, what is therapy? Therapy is nothing but modification, manipulation. So when you modify your genes, is it in something that we were discussing earlier also, gene editing? So it's the same only. Now, whenever we talk about gene therapy, basically you say it is of what can you modify? Either you can modify somatic cells, which is basically non reproductive cells, which is basically all your cells apart from the cells found in your reproductive parts, which is your sperm and the egg portion. Okay. So basically, all other cells of the body, those are non reproductive, they only go through mitosis, which is your asexual reproduction. But the sperm cell, the egg cells, they undergo meiosis. Meiosis is your sexual reproduction. In mitosis, you have only two daughter cells from one cell. That means one cell divides to give you two cells. Okay. And in meiosis, you have four cells getting formed. Okay. don't worry. What we'll do is in agriculture portion, the next topic that we'll be taking is Mendelian genetics and this meiosis and mitosis only. Why am I taking this? Because very high chances if they can ask you questions on Mendelian genetics, okay, and the three laws of it, or they can ask you questions that why Gregor Mendel chose garden pea as a object for doing his experiment. They can definitely ask you differences between mitosis and meiosis. Okay, so we are going to take that in agriculture portion in the next class. But for now, just remember there are two kinds of cells in the body. One which are found in your reproductive organs, and other are basically all other kinds of cells. The cells which are found in reproductive organs, they are called as reproductive cells or non-somatic cells. And the cells which are found in your other kinds of organs, okay, these are called as the somatic cells. Okay, now when you are modifying, okay, your genes present in the cells, either you're replacing, you're modifying, whatever you're doing, you're inserting or you're deleting, then it is called as a gene therapy. Okay, so what you will do is, see, given you the explanation, that I've given you the process of it. What is happening is, yeah, what is happening is you had this cell which had a gene which was not functioning. Now, what you did is you basically removed that gene and you added new gene. Okay, and now your cell has started functioning again. This is nothing but gene therapy. It's a humble request. Whenever there is a question, please make these diagrams. Okay, now, when I told you that this gene therapy can work in two kinds of cells, gene therapy can work in two different ways also. Either you can take these cells outside the body and then modify and then insert it back. This is called as in vitro. Okay. What is it called as in vitro? You must have heard about IVF. What is IVF full form? It's in vitro fertilization. So you take out, you basically fuse the sperm and the egg outside the body, and then once the zygote is formed, you insert that back into the women's womb. Okay, so this is IVF. So in vitro means outside the body. When it's vivo, it means inside. And when all this therapy or whatever you're doing, okay, modification you're doing, it is happening inside the body itself, it is called as vivo. So either you can call it vitro versus vivo. Vitro means outside the body, vivo means inside the body, or you can call it ex vivo or in vivo. In vivo means inside the body, ex vivo means outside the body. Understood? Do not get confused. Do not get confused. Okay, it is easy. I'm just telling you, just remember IVF, in vitro fertilization. Just remember that. Forget about in vivo and ex vivo for a while. Just remember in vitro fertilization. In vitro means what? You are fusing, you are fusing outside the body, right? So just remember, modifications are happening outside the body, and vivo means inside the body. Similarly, I told you two kinds of cells you can modify: somatic cells, which means what? Non-reproductive cells. Okay, so that means whatever changes you are making, they will not be inherited. Okay, only the changes made to egg cells or sperm cells, okay, those are inherited because those are your reproductive cells. The changes that you make to any other kind of cells are non-reproductive. That means these are non-inherited. That means the changes will be only present in this generation. Another, again, when the offspring is born, you have to again go through the same treatment and make the same changes. Okay, if you make those changes in the reproductive cells, those are also called as germline cells. Okay, what are they called as? Germline cells as well. And so when the changes are made to germ cells or germline cells, it is called as the germline gene therapy. Okay, then I told you about in vitro and in vivo, and then there is ex vivo and in vivo. In ex vivo, cells are modified outside the body. I told you, for example, your CAR T cells are taken outside, then modified, and then inserted back. In vivo means within the patient's body only. For example, when you're treating viruses. Okay. Now tell me, do I really need to, tell you the applications of gene therapy? Obviously not, because now you must have gotten the idea. What you can treat your leukemia, cancer, CAR T cell therapy, just your thalassemia, all these genetic diseases also, cancer treatment, your COVID-19 like virus treatment, okay, your HIV treatment. Similarly, you will find there are other disorders and diseases. For example, this Parkinson's disease, which is present in your aging people, your spinal muscular atrophy. So these are the disorders that you find generally in your old age. And why in old age they're happening? Because those genes are regressing. Okay, those genes are basically losing their functionality. So when you cure that functionality, okay, using this gene therapy, what you're doing, you're making them active again. Okay. If you talk about gene therapy products which have been approved in India and which are in use, okay, you will find that in India, I already told you to remember this name, okay, which is Zolgensma, and this was approved in 2022 only for treatment of this particular disorder, spinal muscular atrophy. Okay, in US, you will find that there are many more, products which have been approved. In India, again, this next car 19, which is that IIT Bombay and Tata Memorial services combination where they are using CAR T cell therapy. Okay, amazing. Now let's come to the another concept, which is genome sequencing. What I have done in this classes, I've already discussed, okay, major pointers about each of these earlier only, so that whenever we are discussing this doc, it becomes easy. Okay, it doesn't, it doesn't becomes very difficult or grasp, or as a very difficult to grasp, or a concept which you feel you cannot understand. Genome sequencing, nothing. But I told you these nitrogenous base pairs are there. How are they ordered? Whether it is ATGC, whether it is ACGT, whether it is AGCT. So the ordering of this particular nucleotide sequence, okay, this is what we want to find out when we're talking about genome sequencing, okay, or DNA sequencing. Understood? So this exact order you want to find out. Now, again, see, you must have heard that our DNA contains somewhere around 3 million base pairs. That means 3 million of these ATGC. So, do you want to find out the entire 3 billion base pair sequence? Then it is called as whole genome sequencing. Okay. You just want to find out a particular gene sequence, then it is called as the targeted gene sequencing. Okay. Or you want to find out the exon sequencing, which is basically the genes also have but different parts. Okay, some parts are codified to produce proteins, okay, and some parts are not. So when you just want to find out the sequencing of the part which produces proteins, then it is called as the whole exome sequencing. Okay, why am I giving you the types? Because this can be asked as a question. Explain genome sequencing, explain its types, its significance, and applications. So genome sequencing, you already know, identifying the sequence of these base pairs or nucleotides in our DNA. Types 3, when you want to find out the entire genome sequencing or 3 million base pairs, when you only want to find out a particular gene sequencing or that part of gene which is coding for proteins. Okay. Significance, obviously, if you know, you can identify a particular characteristic, you can give them personalized treatment, you can identify that why a disease is happening in a particular location. You can also, produce personalized treatments. You can basically cure some genetic diseases. You can also do research, agriculture research also, because you can do this for crops as well. You can do this for animals also. You can revive dire wolves, remember. So all this can be a part of genome sequencing. In India, there are two programs I had already told you. One is being run by CSIR and it is called as IndiGen program, where you are finding, you're sequencing 1,000 Indian individuals. Basically, it's 1,008, that holy number. Okay. So 1,008 individuals you are trying to sequence their genome in in indigen program, which is run by CSIR. And then there is another program which is called as your Genome India project, which is by IISc Bangalore, in which you are sequencing 10,000 Indian people. So basically what we're trying to do is we want to find out that why a particular disease is happening in a particular population, what characteristics are there, how geography is impacting those characteristics. So obviously you need a much larger database, and hence 10,000 individuals. Applications, I already told you. So significance and application is almost same. Okay, you talk about, you take different fields and you start, basically writing the uses of it. Okay. Then comes the another part, which is your stem cell and stem cell therapy. I've already told you, whenever you hear the word stem cell, always remember the picture of zygote. That how a small tiny nucleus, which is just formed after fusion of your sperm and egg, and now it is dividing to give you an entire individual. That means that initial amount of cells, okay, they are giving you different kinds of cells. Okay, they are dividing to give you liver cells, lung cell, heart cell, uh, every different kinds of cells. Okay, so this is your stem cell. Now what is happening is, suppose there is an individual, right, like me or you. Okay, we have heart cells, blood cells, etc. There are different kinds of stem cells. Okay, stem cells which can give you any other kind of stem cells. So those are basically your embryonic stem cells. Okay, similarly, you will find there are adult stem cells. Adult stem cells means which only perform one particular function. Okay, for example, brain cells will perform only brain functions, heart cell will perform only heart functions. Similarly, you will find there are induced pluripotent stem cells. Very, very important. This is what we use for our research purpose. What is induced pluripotent stem cell? These are the cells where adult stem cells have been modified, okay, to behave like pluripotent stem cells. Okay, they are now behaving like pluripotent stem cells. That means you took out brain cells, you modified it, and now brain cells can divide and differentiate into heart cells, liver cells, etc. So, idea is using adult stem cells, using gene modification, now you're producing different kinds of cells. So, such cells are called as induced pluripotent stem cells. Okay, understood? Now, what is the significance? Obviously, what will you do of stem cell? Suppose your liver is not functioning. Okay, you took, for example, brain adult stem cell. You basically induced pluripotency. You supposed to brain cell. You induced pluripotency in this, and now this brain cell can divide into your heart cell, right, or your liver cell, and that is how your liver can start functioning again. See, if your liver is not functioning, basically it means what? Your cells are not functioning. So if you have well functioning cells again, your liver will start functioning. So what is it doing is stem cell therapy is can be used for treating a lot of disorders. For example, leukemia, thalassemia. It can be used for producing medicines also. It can be used to repair or damage tissues and organs. It can be used to produce different kinds of drugs, right? For animals, etc. as well. And lastly, it can be used for understanding your humans. Instead of carrying out clinical trials on live humans, now you can just suppose if you're developing a brain medicine, what you can do is you can produce a brain like organ from stem cell and basically check the efficacy of your medicine. Isn't it helpful in research also? Obviously, yes, right? Challenges again the same: ethical concerns, fund, functionaries, skill, regulatory, okay, awareness, inequality, all that. Okay, and why are we discussing stem cell again? Because in India, recently, Delhi High Court has given the permission for using stem cell therapy for what? Treatment of autism spectrum disorder. Okay, understood? So that is why we are discussing. So here, this is the diagram that you can make when you are, writing in your answer copies also. First, what you do is you basically suppose this is a patient, okay, whose some organ is not working, and now you need some stem cells from it. So what you will do is you will take, suppose that you took out their blood cells, okay, you collected the blood cells from the patient's body. Then what you did is you basically took these stem cells from it, okay. Then now you made some modifications in the stem cells, you infuse those stem cells back into the patient's body, and now those stem cells are functioning well and they are producing that particular organ that you need. Okay, now okay, two words that I want to explain is one is yeah, here there are two words, one is parthenocarpy and one is parthenogenesis. Almost the same, but the context is different. Sometimes you must have heard that there is a mango, okay, which is seedless. It doesn't have a seed, that gutli inside it. Okay. So basically, generally, those kinds of fruits are produced from parthenocarpy. Parthenocarpy means generally a fruit is produced from what? A seed. A seed is made by fusion of pollen grains and your style ovary. Remember, in flowers, you have this stamen, pistil, style, ovary, ovule. Okay. So you have your female gametes and your male gametes. When they fuse together, they give you seed. And from the seed, your fruit is developed. Okay. So your mango is developed from the mango seed, which is getting developed from the fusion of your male and female gametes. Okay. Sometimes a fruit is developed without this fusion process. That means there is no male or female gametes fusion taking place. Okay, directly the fruit is getting produced. So that process is called as parthenocarpy. Now extend that to humans or animals. When uh, an organism is formed not from the fusion of male and female gamete, but directly from the egg. That is called as parthenogenesis. So it is a type of asexual reproduction, obviously, because sexual means what? There should be fusion of male and female gamete. That is not happening. You had an egg, and from the egg directly you will find there is development of a new organism in itself. Okay, the whole new organism can be developed without fusing it with the male gamete. Okay, now, okay, so why am I telling you parthenogenesis? Because recently parthenocarpy and parthenogenesis both were in news. Okay, Wo- I so I think you will be able to understand parthenocarpy and parthenogenesis now. Okay, so do not get confused. Whenever you hear the word carpy, it means AR, that means agriculture. And when you hear the word parthenogenesis, it is basically talking about humans and animals. Okay, remember, in humans, we do not observe this process of parthenogenesis. If by chance you had a huge like you were a fan of watching these web series, you might have watched Doctor Who series, okay, and House. In that House, there is one patient wherein the lady gets pregnant, and she says that she did not had sexual relations with anybody, so how can she be pregnant? And that's when House discovers that, okay, you are the first case in the world of parthenogenesis. Okay, so I am just giving you a like a web series example over here, but the point is that this has not been observed in humans so far, but this is very common in lizards, alligators, and all these insects, specifically insects, bacteria. This is a very common process wherein without fusion only, they basically asexually reproduce, and an entire organism is just formed from the female gamete itself. Okay, so it can be occurring naturally also, and it can occur artificially as well. In when you do cloning and when you do research in the labs. Okay, but do not mix it up with the parthenocarpy. So now this was a very, very long lecture, I know, but your entire biotech portion, all those technologies which were in news have been discussed, and you don't need to worry about biotech portion. If you have any feedback, you're most welcome to mention it, and we will try to incorporate that in the next video. Thank you, and hope to see you again. Thank you.